You've seen in the previous video how proteins are targeted to the endoplasmic reticulum, specifically focusing on soluble proteins that will end up in the ER lumen. I want to briefly review that, but focus this video on proteins that are targeted to the endoplasmic reticulum membrane. As a review, recall that ribosomes begin synthesizing proteins by reading mRNAs, and if the synthesized protein contains an ER signal sequence at its extreme amino terminus, translation of that mRNA is halted, and the complex of mRNA, intact ribosome, and partially synthesized protein is targeted to the endoplasmic reticulum, where translation will be reinitiated and completed. This is what we term rough endoplasmic reticulum, but I would like you to consider, is there such a thing as rough endoplasmic reticulum? Is an endoplasmic reticulum always rough, or is it sometimes smooth? This is the ER targeting sequence, and remember that we are going to have a string of at least 10 consecutive hydrophobic amino acids that's going to form an alpha helix, and that will be preceded by at least one positively charged lysine amino acid. Again, as the mRNA is being translated, that ER signal sequence will be recognized in the cytoplasm by SRP, signal recognition particle, and binding of SRP to that ER targeting sequence will cause the ribosome to halt translation until the complex is able to bind to an SRP receptor in the ER membrane. The SRP receptor will transfer the complex to a translocator protein, and when that occurs, the SRP is displaced because of GTP hydrolysis, and at that point, translation can continue. Translation will occur through that translocator channel such that the newly synthesized protein is being translated through that pore into the ER lumen. Of course, that continues until translation is completed. At some point during translation or at the end of translation, an enzyme known as signal peptidase will cleave at the end of that hydrophobic amino terminal sequence, leaving the signal peptide embedded in the ER membrane and releasing the soluble protein into the ER lumen. Now I want to focus on transmembrane proteins. Any transmembrane protein that is delivered to the endoplasmic reticulum will complete its synthesis there, but remember that protein synthesized at the ER will ultimately travel through the secretory pathway. Therefore, a transmembrane protein that is synthesized at the ER can end up anywhere within the secretory pathway. So we're talking about transmembrane proteins that might be retained in the ER, might be retained in the Golgi, might be delivered to the lysosome, or ultimately end up at the plasma membrane. We'll start with the simplest type of transmembrane proteins, and these are what we call single-pass transmembrane proteins. They pass through the membrane a single time. There are two orientations that a single-pass transmembrane protein can have, and this slide depicts one of those two ways. That is, a protein can have two hydrophobic stretches of at least 10 amino acids in length, and the first of those will be found at the extreme amino terminus, representing the signal peptide that will be responsible for delivering the protein to the endoplasmic reticulum. That signal peptide is what we're going to call the first hydrophobic starch transfer sequence. That means that when the ribosome is delivered to the translocator pore at the ER membrane, translation will begin to be reinitiated, and the protein that's being synthesized will begin to be transferred through that translocator pore. Upon reaching or translating a second hydrophobic stretch of 10 or more consecutive hydrophobic amino acids, transfer through the translocator pore will stop, although translation will continue. That results upon cleavage of the signal peptide in a protein whose amino terminus is within the lumen and whose carboxyl terminal end is in the cytoplasm. The second option is for the protein to be oriented in the opposite direction, with the N terminus in the cytoplasm. This can and does occur, but it cannot happen if the signal sequence or the signal peptide is at the extreme amino terminal end of the protein. Instead, the ribosome will translate the mRNA and translation will continue in the cytoplasm until a signal sequence, again a hydrophobic stretch, in this case surrounded by that lysine amino acid, will be recognized by SRP. SRP will bind to this internal hydrophobic signal sequence, stop translation, carry the partially synthesized protein and the ribosome and mRNA complex to the ER, just like it would if the signal sequence were at the amino terminal end. Translation stops until the complex is delivered to the translocator pore, at which point GTP is hydrolyzed, SRP is released, and translation continues through the translocator pore. In this way, the carboxyl terminal end of the protein is in the ER, and the amino terminal end remains associated with the cytoplasmic face of the ER membrane. Looking at how these proteins would appear in a sort of linear fashion, the blue line or the blue rectangles represent the protein and the red/orange represent the hydrophobic stretches. A single pass transmembrane protein with its amino terminal end would have two hydrophobic stretches in the protein, one at the extreme amino terminal end and one internally. Of course, the one at the extreme amino terminal end will be cleaved by signal peptidase, creating a new amino terminal end that is not hydrophobic. The second scenario demonstrates a single pass transmembrane protein with the amino terminal end in the cytoplasm and the carboxyl terminal end in the ER lumen. In this case, the protein would have only a single hydrophobic stretch, and that hydrophobic stretch will be internal. More complex than single-pass transmembrane proteins are multi-pass transmembrane proteins. These are proteins that pass through the membrane multiple times. In this image, we're looking at ultimately a two-pass transmembrane protein with both the amino terminal and carboxyl terminal ends on the cytoplasmic side of the ER membrane. In this case, there are two hydrophobic stretches in the protein, both internal. When the ribosome reaches the first of those, the one that's colored red here, first of those hydrophobic stretches in translation will stop. This will be analogous to the signal sequence SRP will bind and carry the complex to the ER membrane, transfer the complex to the translocator channel, and translation will continue through the translocator channel until the second hydrophobic stretch, in this slide labeled the stop transfer sequence, is reached. When that sequence is reached, transfer will stop, but translation will continue to the end of the coding region of the mRNA. At that point, translation will stop and the carboxyl terminal end will remain in the cytoplasm. The way that this protein would look in a linear fashion is shown here. Again, the blue being the protein and the two red and orange colored sections being hydrophobic stretches. Of course, our cells have multi-pass transmembrane proteins that weave through the membrane more than just 2 times. In order for us to look at an amino acid sequence and predict the orientation of a protein within a membrane, we can use what is shown here, a hydropathy plot. Looking at the amino acid sequence of a protein, we can look at regions of the protein that are stretches of hydrophobic amino acids, and this may indicate that these hydrophobic stretches are indeed transmembrane stretches. This process of using hydropathy plots to predict how a membrane or how a protein might be oriented in a membrane is known as membrane topology. If we look at this protein, the one that is shown in the hydropathy plot, in a linear fashion like we had previously, we see that there is a hydrophobic stretch at the extreme amino terminus, and then there's a hydrophilic region, then another second hydrophobic region, etc., etc. We know that the hydrophobic stretch at the extreme amino terminal end will be cleaved by signal peptidase when that amino terminal end is in the ER lumen. So that will not be in the mature protein. The new amino terminal end will be luminal. This is not a surprise because also shown in this diagram are these sugars, carbohydrates. We know that carbohydrates are placed on proteins in the ER lumen, so this tells us that the new amino terminal end will be in fact luminal. Each of the hydrophobic stretches can be a transmembrane region, and if we were to diagram this protein in a membrane, we would see that each of the green sections would alternate luminal and cytoplasmic. Let's look at some other examples. In the top example, we've got an extreme amino terminal hydrophobic stretch, a green hydrophilic stretch, followed by a red hydrophobic stretch, etc., etc. If we were to draw each of these proteins in the membrane, of course, labeling which sides are cytoplasmic and which sides are in the lumen of the ER, we can know that the hydrophobic stretches might be embedded in the membrane. With the top example, the extreme amino terminal hydrophobic stretch would be cleaved by signal peptidase, and then all we do is draw in the remainder of the protein, the hydrophilic stretches. Our membrane topology would look like this. The new amino terminal end would be luminal, and the carboxyl terminal end would also be luminal. In the second example, we don't have an extreme amino terminal end. The first hydrophobic stretch is internal. We know then that the amino terminus of the protein will be cytoplasmic. We draw each of the hydrophobic stretches in the membrane. And we draw the rest of the protein in the membrane. Again, in this case, the amino terminal side of the protein in the cytoplasm and the carboxyl terminal end of the protein in the lumen. We'll have more examples like this in class, but this is a mechanism by which we can predict protein topology within a membrane for multi-pass transmembrane proteins.